Hello everyone, welcome to today's live broadcast. Run your assays faster, getting the quantitative answers you need for N-Glycan, CE-SDS, and charge heterogeneity applications without the wait. I'm Robert Castellanos of Labroots, and I'll be a moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by SCIEX. SCIEX helps to improve the world we live in by enabling scientists and laboratory analysts to find answers to complex analytical challenges they face. SCIEX global leadership and world-class service and support, and the capillary electrophoresis and liquid chromatography mass spectrometry industry have made it a trusted partner to thousands of scientists and laboratory analysts worldwide who are focused on basic research, drug discovery and development, food and environmental testing, forensics, and clinical research. For more information, please visit SCIEX.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event, as well as responding to poll questions that will be asked. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the C button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would like to introduce today's speaker, Matthew Salem, followed by Dr. Mark Lies, who will be addressing the questions during the Q&A portion. Matt received his bachelor's degree in chemistry from the University of California at Davis, focusing primarily on analytical chemistry techniques. He went on to get a master's of biotechnology degree from San Jose State University, during which he took both science and MBA level business courses. He started his career at Agilent Technologies, working with the gas chromatography teams, doing content marketing before eventually managing the marketing programs for the GC/MS product lines. After moving to the separations group within SCIEX, Matt began managing the marketing for the standalone capillary electrophoresis products, including the PA800+ instrument. In his role, he is dedicated to driving the adoption for CE for biologic characterization as a highly quantitative orthogonal technique to a more traditional HPLC-based methods. Dr. Mark Lies is a global business manager for the PA800+ at the SCIEX Separations in Brea, California. He is focused on driving the adoption of CE-based assays as a valuable asset and orthogonal data set, traditional LC methods, in both the biopharma industry as well as the academic research sites throughout the world. Prior to that, he worked on the global marketing manager at Beckman Coulter before CE products were moved to SCIEX, promoting the advancements in CE technology over the years. Dr. Lies attained his PhD in molecular genetics from the University of Kentucky after completing a bachelor and master's degree in the program in the biological sciences at the University of Massachusetts at Lowell. His core work at UKY included the characterization of Analyses promoting complex Aspergillus sp. I'll now turn it over to Matthew for his presentation.
Thank you very much for the introduction, Robert. And thank you everyone for joining us. Today we've got a presentation on fast quantitative results for N-Glycan, CE-SDS, and charge heterogeneity applications. So just a quick look at the agenda for this morning. As I mentioned, we've got 3 assays that we want to go through, but first I want to spend just a little bit of time discussing the framework for all of this, which is the importance of quantitation. So I'll spend just a couple of minutes walking through this section on quantitation when performing characterization assays on any of your molecules, and why it's important to remember that just because we're going faster doesn't mean we're going to try to lose any of that quantitative data. So in general, we've got 2 buckets of data, qualitative and quantitative. The qualitative bucket is usually where we see faster separations to give you those quick answers. You're generally looking for higher throughput, and it's used earlier in the development cycle. It's really just meant to give you those sort of go, no-go decisions. On the other side, we have quantitative data. Quantitative data requires a couple of things, namely high resolution, high sensitivity, and highly reproducible assays. The reason for that is that these are often used for FDA submissions closer to launching a product, or even just later in the development cycle, so that you can really have confidence in the results that you're getting. Now, we mentioned that fast separations normally falls into that qualitative bucket, and that's the theme of today's talk, is fast separations. But what we've done is actually optimized our methods to stay within this quantitative data set. So the same data set that you've been accustomed to getting on the P800 Plus, you can still get using these applications. We've optimized the method to make them a little bit faster. So just a little more proof behind the quantitation. So I mentioned that high resolution and high sensitivity are very important for us, especially towards the later stages of development. And this is just some CE-SDS UV data showing 3 logs of dynamic range. So we've got our 10 kD protein in there on the left, and you can see we can get detection down to 0.1% lysozyme. The graph on the right, again, just proving 3 logs of dynamic range. Now again, this is with UV analysis. If you needed higher sensitivity and you needed to push the instrument just a little bit farther, we can use LIF detection, laser-induced fluorescence, and get a little more sensitivity out of it for any assay where you may need to get down to closer to 0.01% impurities. Another thing I mentioned when talking about quantitative data is this idea of reproducibility. And for capillary electrophoresis, one of the biggest factors of reproducibility is capillary temperature. So here we are taking a look at the effective temperature on migration times. And really the only way to accurately control what the migration times are going to be run to run is to very precisely control the temperature in the capillary. And we do that by circulating cool liquid over the capillary to really precisely control that temperature. So the graph on the left shows you a separation done at 20 degrees C, and you can see our standard ladder sort of at the top and our first peak down there coming out at about 6 minutes on the bottom. Now if we move that up to 50 degrees C, you can see a shift. So you're obviously going to start running the assay a little bit faster because as you increase temperature, things are moving a little bit faster. And so that first peak then comes out at about 4 minutes. And while that might be interesting to some people to run your applications a little bit faster, you lose that reproducibility if you can't actually control the capillary temperature. And so it's very, very important for us to make sure that when we are looking at quantitative data sets that we can actually have very low percent RSEs for both migration time and corrected peak area. And so liquid capillary cooling is a major factor in all of that. The last piece here on quantitation is just some studies that have been done on the various applications that we'll talk about today. So these were multi-site studies, so intercompany collaborations between biopharma companies to validate P800 Plus applications. It started with CE-SDS back in 2006, and then they moved on to charge heterogeneity, looking at CEIF applications in 2011, and capillary zone electrophoresis, or CZE, in 2014, and most recently at the very beginning of this year, examining N-Glycan analysis. And I've got the references for all of those papers down below if anybody's interested in reading those. Again, just more proof from industry partners and collaborators that these assays are reliable, they're robust, and they're quantitative. No matter where you are in the world, whether you're porting methods from Europe or Asia or any other part of the world, we can maintain that quantitative data across sites and across analysts. So moving on in the agenda, so we've talked about the importance of quantitation, and again, even though we're going to talk about fast applications, we're not necessarily going to talk about losing quantitation. The first one is N-Glycan analysis, and a poll should pop up on your screen, and we would like to get your input on how you do N-Glycan analysis today. So I think we'll give people just one more second to go ahead and answer the poll. Okay, so it looks like a fair number of you, actually 3/4, are not doing N-Glycan analysis at this time. So we'll run through N-Glycan analysis by CE. It looks like there are a number of you that use HPLC or HILIC methods. And I'll talk through each of those and the advantages of using CE for glycan analysis. So for those of you that maybe aren't doing N-Glycan analysis now or aren't familiar with glycosylation analysis, I just wanted to sort of, again, frame the conversation around why we care and why we do this. So there are a number of challenges for N-Glycan analysis, but we're seeing a growing demand for glycosylation profiling of therapeutic antibodies. The reason that they're challenging is because they've got a lot of unique physiochemical properties. They're very complex and diverse. For CE applications, there's a lack of charge, which makes the separation difficult. And there's really no chromophore or fluorophore that lets us detect these things easily. So a derivatization step will be involved, and I'll explain that in a minute. I mentioned that they're very complex, and the oligosaccharide expression with over 600 enzymes that are responsible for glycosylation. So there's a high degree of variability, which means that understanding the glycosylation on your monoclonal antibody is very, very important. The high level of structural diversity and the extensive sample prep add to those. So extensive sample prep could be days in some cases if you're using certain HPLC methods. And we've optimized our sample prep as well, so I'll go over that in a minute. But in general, glycan analysis is very difficult, but we're seeing a demand in the industry for it, and so we've really tried to optimize our methods to make it easier and faster for everybody. So this is just a quick look at where we're going to go. We've got this really fast CE-LIF analysis that we can do for N-Glycans, and it's going to give you both a high-resolution analysis and identification of all of your peaks in just a couple of minutes. You can see the electrophoregram here, the run with the final standard coming out, DP15 there, at about 6.5 minutes. So you can run your whole sample in under 7 minutes, and you can get quantitative results and peak identification at the same time. So that's kind of the proof point. That's where we're going. But how did we actually get there? So I mentioned sample prep can be a bit difficult for glycans, and that's usually true. But we've optimized our sample prep method to get everything done in 60 minutes. So you can go from adding your sample to the vial to eluting it with water and being able to inject it into the system in just an hour. So I'll walk you through just the key main steps here. We use a magnetic bead-based sample prep, so you add the beads in your sample, a quick denaturation, and then a PDFF digestion to cleave off the glycans. We pull the glycans down and attach them to the magnetic beads, where we can do APTS labeling, and APTS is just the label that we use. I'll go over that in more depth in a slide in just a minute. After the labeling, we do just a quick cleanup to wash out all of the excess APTS, and then we elute the glycans off of the magnetic beads with water, and you have your sample, and it's ready for CE-LIF analysis. So start to finish, your sample prep for up to 96 samples can all be done in just an hour. Now, I mentioned 96 samples. If you have a need for higher throughput for your glycans, we do recommend some kind of sample prep automation. And again, because there's no centrifugation steps like you might have with some HPLC methods, we can automate the entire sample prep process, meaning you can do 96 samples in that same 1 hour timeline. Automation is also nice because it eliminates potential operator error, normal pipetting errors, that sort of thing. And obviously it increases lab throughput and efficiency if you can run through more samples quicker. So if you have a throughput challenge, we definitely recommend automation. So moving on past the sample prep, or I guess to a very specific part of the sample prep, I mentioned that we have to label our glycans with APTS, and the reason for that is twofold. One, to introduce charge so that we can actually do an electrophoretic separation, and a fluorophore so that we can actually detect things with laser-induced fluorescence. So the label that we use in our sample prep is APTS. It's an aminopyrene trisulfonic acid, and it adds both that charge and that fluorophore. It's a very simple and efficient reaction. It's just a simple reductive emanation, single step, and it's got over a 90% labeling efficiency. It is non-selective, so you get uniform labeling, and it makes it very easy to quantify because you're only labeling the reducing ends of the sugar only. So you get one fluorophore per glycan molecule. So when you actually detect these things, the quantitation becomes exceedingly easy. And the nice thing about using LIF detection is that it's very, very sensitive. So you can get orders of magnitude of linear range using the LIF. So along with the sample prep, moving into more the actual analysis, the separation of the glycans, we've developed this high-resolution N-Glycan separation gel for the capillary, which we refer to as our HR-NCHO gel internally. The gel is optimized to increase resolution without increasing separation times. So you can see here we've got 2 electropherograms. The one on the left is just your standard carbohydrate mix with all of its constituents down below. And you can see the time frame down there at the bottom. These things are all coming out under 5.5 minutes. So again, very, very quick separations, but still very good resolutions. If you move over to the right-hand side, you're looking at a more real-world sample. This is a human IgG glycans. And again, you can see the time scale down at the bottom. Everything is getting resolved in less than 5.5 minutes. And again, baseline resolution for most of these species, we get still very high resolution even though we've increased the separation time significantly, allowing you to run your glycans even faster. You will notice at the top of both of these, there's a couple of numbers, G4, G5, G6, etc. I want to walk through what those are because those are very important for the way that we quantitate and identify what our peaks are in the software. So those G values that you saw running across the top of those last couple electropherograms are what are called glucose unit values, GU values for short. GU values are an abstract unit to express migration time. So they basically are just a way for looking at incremental glucose additives and how long it takes to separate those. So G1 would correspond to 1 glucose unit, G2, 2 glucose units, etc. So you basically build a standard ladder almost that you can then reference against your human IgG glycans, for example. The reason we use GU values is to predict exoglycosidase digest shifts. And by doing so, we can use a database that actually provides immediate glycan identification based on the relative migration time between your sample and the GU values that are known. Another nice thing is that it's instrument independent. It's not something that you can't port from site to site. It's very easy to use, and GU values are actually automatically assigned within the P800 Plus software. So as you're running your glycans, it will look at the relative migration time from a number of internal standards that we use, and those will be used to actually assign the GU values. So I explained that a little bit more in depth here. GU determination and glycan identification. So if you look at the very first peak on the far left where it says GU2, that's actually one of our standards. And we spike that in with the sample as we do the analysis. And the relative migration time for all of those peaks that you see versus that standard is what's going to give us these GU values. And once we get the GU values that you see here on the screen, like 7.62 or 8.70, we can do a database query, actually match those GU values with a predicted glycan structure. So now you've not only just gotten your separation done in 5 or 6 minutes, but you've also identified what your glycan species are for each of those major peaks. So it's not just the separation, but we can also do identification in just a couple of minutes using one single method on one instrument. The real benefit to this, for those of you out there that are using HILIC, is that you can run your glycans up to 5 times faster on CE using our method than previous HILIC methods. You get that high resolution glycan data, right, and identification in just a couple of minutes. And it's from start to finish, right? We've optimized every step of the process. So sample prep in just 60 minutes for, again, up to 96 samples. Separations in sometimes less than 7 minutes, depending on your sample. And high resolution data and automatic identification from within the software. So we've got a table here at the bottom that just gives a quick comparison. Sample prep for HILIC can take days in some cases, but for the most part, methods that people use are 3 hours. Separation times, if you're looking to get the same kind of resolution and sensitivity that we get on CE, you generally have to run about a 45-minute separation. Conventional CE, this is sort of the way we used to do it. The sample prep used to take about 4 hours, but we had a separation time of about 20 minutes. We've increased both of those with this fast glycan method to, again, be 1 hour for sample prep and 7 minutes for the separation. So you get a huge time saving here. So I want to go back to this electropherogram that I showed you at the start of this glycan section, and now hopefully it makes a little more sense how we got here. So again, the separation down at the bottom, you can see that we get everything out in under 7 minutes. The DP2, DP3 peaks at the beginning and the DP15 peak are our triple internal standard that we use for this analysis. And again, the relative migration time between those standards and the peaks that actually come out on the electropherogram are what we use to assign GU values. And those GU values are then matched against the database to give us immediate identification of each of those peaks. So that's how we can label all of these with highly reproducible migration times and be able to tell exactly what they are. I mentioned reproducibility. Obviously, if we're trying to accurately identify all of our glycan species, we can't have things coming out at different times, right? So reproducibility for migration time and corrected peak area are very, very important. So here's just some representative data actually showing the RSDs for corrected peak area down at the bottom in that sort of red box. Everything is less than 3% for our key glycan species, and a number of them there are even below, well below 1%. So very reproducible data. Again, not just fast for fast's sake, it's fast quantitative data that we get. It's very reproducible. for our glycan analysis. Now, we've talked about glycans and the fast analysis, and all of this was made possible by this kit that we've developed, this fast glycan labeling and analysis kit, which is coming very soon. We have a new part number for a kit. If anybody is familiar with our current carbohydrate labeling and analysis kit, this will be a separate kit. It comes with everything you need to do the analysis, with the exception of the P-enginease F, and we do that just to give people flexibility on the enzyme that they use to cleave the glycans. But the kit comes with the high-resolution glycan separation gel, which I talked about earlier. It comes with a separation capillary and a magnetic separator for the sample prep, and then all of the solvents and standards that you would need to actually run the assay. Along with the kit, we're going to be shipping a pre-assembled capillary cartridge with every kit. So it's going to basically increase your uptime. You won't have to wait as long. You won't have to worry about threading your capillaries or cutting the tips just right. We'll send you a pre-built cartridge that you can use for this analysis. So again, this is a new kit we're coming out with. We should be releasing in the next couple of months. If you're interested, you can obviously let us know. And it's going to have everything you need to do all of this fast glycan analysis that I've just discussed. Switching gears slightly, but staying on the topic of fast applications, we wanted to talk about fast CE-SDS applications. We've got a very interesting optimized method that lets you run 2x faster than our standard CE-SDS method and 2 times higher throughput, just optimizing the tray configuration. And then I'll go into a little more detail about those pre-assembled cartridges because you can use those for the SDS analysis as well. I think we have another poll here that's going to discuss how you actually do SDS analysis if you're doing that today. So we'll give people just a couple of minutes to answer that poll. If you are currently performing SDS analysis, how do you do it? Using the P800 Plus system, another CE system, or the standard slab gels, gel electrophoresis, or if you're not doing it, not at this time. I think we'll give everybody just one more second to answer the poll before we close it. Perfect. Looks like there are a fair number of you, almost a quarter of you out there that are doing CE-SDS analysis on a P800 Plus. That's great. And I assume most of you are using our standard method. If you are, then you'll be very interested, hopefully, to learn about the optimizations that we've made to that method to increase both your speed and your throughput. So let's walk through the CE-SDS analysis. So we've got 2 sample analysis methods for CE-SDS, and both, as we'll see later, can be configured with our high throughput tray setup, but I'll get to that in a minute. The standard method that those of you who are running on a P800 Plus today are probably familiar with takes about 30 minutes. You've got about a 30-minute separation time with a 20-centimeter effective length capillary. Anybody that's not familiar, effective length is the point of injection to the detection window. You're running in a left to right capillary configuration, which you can see in that sort of diagram down at the bottom. The standard CE-SDS method injection runs up the left side of the capillary through that long distance, the 20-centimeter distance, through the detection window and then into the outlet vials. Using the standard method, you can run about 48 samples per day if you're constantly changing, if you make sure you set up the trays correctly. With the high-speed method, however, we're going to cut the separation time in half. So it was 30 minutes with the standard method. It's now 15 minutes on the high-speed method, and that's because we're cutting effectively the capillary length that we're using. So we're actually running up the short side of the capillary, so that 10 centimeter effective length part of the capillary, running right to left. So we're basically just switching the inlet and outlet trays in the system and running it in reverse, basically. The nice thing about this, not only can you run faster, so you're going to cut your separation time in half, but you can also increase the number of samples you can do per day. So your throughput goes up to about 72 samples per day, up from 48 previously with the standard method. And again, this is just a method optimization. The kit that you see down there at the bottom is the same CE-SDS kit, whether you're doing molecular weight or IgG purity, heterogeneity. It's the same kit. There's nothing new. It's just an optimized method. We've been working very, very hard to make sure that we haven't changed, or we've changed very little, just enough to get us higher speed and higher throughput on the assay. So this is what some of the data looks like for the standard ladder. So you can see on the top is the high-speed method with the 15-minute separation coming out. It looks like between 14 and 15 minutes there. And then on the bottom is that same ladder run on the standard method, where you see it coming out closer to 28 minutes. You still get very, very good resolution on the high-speed method, even though we are running it faster. But obviously you get the benefit of being able to run more samples if you're cutting your separation time in half. One more set of data to pit the high-speed method versus the standard method. So the graph here is showing standard, which is denoted with an S on the left-hand side, and the high-speed method, which is denoted by HS on the left-hand side. We're looking at both reduced and non-reduced analysis for CE-SDS. And you can see, again, the time change is significant. So for the reduced, we're actually coming out right around 11 minutes for the high-speed method, whereas we're at right around 20 minutes for the standard method. But you still get that non-glycosylated peak there. Again, still good sensitivity and resolution on things like the light chain and the heavy chain. And even with the non-reduced, we still get that non-glycosylated peak there. But again, with a significant increase in, or decrease, as it were, in separation time. So as I mentioned before, all of this is just method optimization. We haven't, tried to develop a new kit to make it run faster. We've just done some really interesting applications work to make sure that this works. And it's all outlined in a tech note that we have on our site. It walks you through everything from the high-speed method itself, running up the short side of the capillary and making sure that you've switched the inlet and the outlet buffer vials, vial trays correctly. It walks through how to set everything up in the software so that the system knows where all the vials are, and you don't have any issues running it basically in reverse. It also walks through the high throughput tray setup. And I want to take a second just to touch on what this tray setup is. Personally, this is probably my favorite part of the method because it seems so simple, yet it took us a long time to really optimize this and figure it out and get it working perfectly. So we used to be able to run 48 samples, we used to be able to run 24 samples on the conventional tray setup that we used with our standard CE-SDS methods before you had to change out reagents. With the high throughput tray setup, we can now double that and run 48 samples without changing reagents. So again, all we've done is sat down and really thought about optimizing the way that all of the vials are placed in the trays so that we don't have to intervene after 24 samples and change out our water and our waste and our washes. Now we can do that after 48 samples. And the nice thing about this tray setup is that it works for either method. So whether you're doing the standard CE-SDS method or the high-speed method, you can use this high-throughput tray setup. And again, effectively all you're doing is switching the inlet and the outlet buffer trays with this new tray setup. And again, all of this is outlined in that tech note. So if you're interested at all, I would invite you to go to SCIEX.com and look up that tech note. It has all of the information that you need in it. I mentioned the pre-assembled capillary cartridges before when we talked about the N-Glycan analysis. We also use them, the same cartridges, for CE-SDS analysis. The nice thing about these pre-assembled capillary cartridges is that they give you uniform, reliable injections every time. They're precision cut. They're actually laser cut so that they eliminate run-to-run variations. If you've had to thread and cut your own capillaries, you know what a big advantage that is. You get very precise window alignment, which reduces failures, increases the robustness of your assays and the reproducibility of your assays. And it's going to save you time and increase productivity if you're not sitting there trying to thread capillaries and cut the edges and etch windows and all of those fun things you have to do with a capillary. So these are available today. You can buy these right on SCIEX.com, I believe. The part number is down there at the bottom if you'd like to give them a try. And again, you can use these either for this CE-SDS analysis or in the future with the fast glycan labeling and analysis kit. So we've talked about a lot. I just want to summarize really quick this CE-SDS section. We've got a faster method, so a high-speed method, which lets you run your samples 2x faster. So we're looking at a 15-minute separation time now. We've also optimized the tray setup, so you can do twice as many samples without having to change reagents, up to 48. And those two things combined give you a true high throughput analysis, right? So we're increasing speed, we're increasing throughput. You can run more samples per day than you ever could before on the system. And we've done all of this, again, without sacrificing the quantitation that we've known and published for years, because we maintain that reproducibility with the liquid capillary cooling, and now with our pre-assembled capillary cartridges. It's much easier. You basically just drop it into the system, set everything up, and you're ready to go. So that is the overview of CE-SDS analysis and the optimizations that we've made there. The last section I want to touch on briefly is charge heterogeneity with capillary zone electrophoresis, or CZE. It can be 2 times faster than our CIF method, and with very, very easy sample prep. As you see, we basically just dilute in water, and simple method optimization. You don't have to worry about all the amphiolites and things that come with CIF. So I think we've got another poll here just to ask about how you do charge heterogeneity analyses today. So there it is. Do you currently perform charge heterogeneity assays? And if you do, how do you do them either on a P800 plus, another CE system, or using HPLC with something like ion exchange chromatography? Or if you're not doing any right now, not at this time. So I think we'll give people just another second before we close the poll. There we go. Looks like we've closed it now. So there are some people that are doing charge heterogeneity analysis today. It looks like probably about a quarter of you, and the rest of you are not. So for those of you that are, we'll walk through the advancements that we've made. You're probably more familiar with CIF. I assume that's probably the application you're using for charge heterogeneity. We're going to talk today again about CZE and how that can be used and why that makes a difference. So again, for anybody that's not familiar with CZE, I want to walk through it just really briefly. It's capillary zone electrophoresis is what that stands for. And really what it is just a mobility-based separation. So you're just separating these molecules based on their charge and hydrodynamic size. The nice thing about it is that you can get sub 10-minute separations. So, between 8 and 10 minutes is pretty typical. It's what you see. I know it's kind of blurry on here, but the graph on the right-hand side, those are all coming out between 8 and 10 minutes. So what happens is you basically just load your sample in, and we'll talk about sample prep in a minute, but after you load your sample into the capillary, it just separates based on hydrodynamic size, so how big it is and how much it's charged. And you get the basic components separated from the acidic components, and you get a nice big main peak usually right in the middle. The nice thing about it, again, talking about reproducibility down in the bottom left with that table, migration time RSDs well below 1%, and corrected area RSDs all below 3%. So we're not losing anything here. We gain some speed over CIEF, but we still get very high reproducibility and good resolution using CZE for charge heterogeneity. So again, just to cover some more about CZE and why it's used or why you might want to start using it as an alternative to CIEF, it's really about speed. So you get high resolution data to characterize your monoclonal antibody, same as you would with CIEF, but you get it much, much faster in, again, sub 10 minutes here most of the time. If you look at the graph here on the right, the time scale is actually at about 14 minutes. The reason for that is actually because in this paper they were using a longer capillary. So obviously a longer capillary, you've got a longer separation distance. The migration times are going to be a little bit longer. But using the standard 30 centimeter capillary, you're going to get separation times on the order of 8 to 10 minutes. The sample prep is another huge bonus for CZE as opposed to CIEF. It's really, really simple. You just dilute your sample in water and inject it into the system. It's pretty much that simple. Again, you don't have to deal with all of the ampholytes and extensive sample prep that comes with CIEF analysis. It's much, much easier to deal with and handle. Because you don't have all of those reagents and you can use a simple bare fused silica capillary, it's also less expensive than running CIEF. And it's, again, highly reproducible, which I showed you the RSDs on the last slide. And method flexibility is much easier. It takes far less time to optimize and develop a method for CZE than it does for CIEF. There's much less tinkering usually that you have to do. Just a quick comparison on CZE versus CIEF. This was actually done by some of our industry collaborators in a series of webinars that I invite you all to go back and watch. The link is down there at the bottom. They basically looked at all of the different ways that you can do charge heterogeneity, and they ranked them basically and wrote down some notes here. So our CIEF, very good resolution, same thing with our CZE, good to very good. The analysis time, again, they were using a bit of a longer capillary length, I believe, which is why you're getting 18 minutes for the separation time on CZE and not the 8 to 10 that I've been telling you about. Applicability to MABs, 100% for CZE. That's because, again, it's an open method, basically. You can optimize it and develop it however you want it based on your molecule. So if you need to change buffers around, you can absolutely do that. It's very, very flexible. Very low buffer consumptions and concentrations that are very, very low. So you use less sample than you would with some other CZE or other charge heterogeneity, I apologize, for CIEF or some other charge heterogeneity applications. So here's a direct comparison between our CZE and CIEF. Again, just to show you, you still get resolution for all three of your main peaks, the basic, the main, and the acidic peak. you get that sort of charge information, charge heterogeneity information in, again, about 10 minutes here as opposed to close to 20 minutes for CEIF. So we talked about CZE a lot and why you would want to use it, but when would you ever need to use CEIF? CEIF obviously still has its place, and it's really when you need that ultra high resolution. So we can get down to 0.03 PI units using our method. It's highly reproducible, and the assay has been proven portable by those multi-site studies that I talked about in the opening. So you get that really high precision, highly accurate PI determination using CEIF. If you don't need that resolution or that really precise PI determination, CZE is a much faster way to get the same information as you would with CEIF. So that's a quick overview of the fast applications that we have. We started with the importance of quantitation. We talked about glycan analysis and how we run that faster with the new kit. We talked about CE-SDS analysis and how we have run that faster with method optimization. And we've shown you an alternative to CEIF for charge heterogeneity that you can run faster using CZE. Now, all of this, all three of those applications are made possible by the system that we run them on, and that system is the P800 Plus. You get automated quantitative analyses, right? We talked about 3 logs of dynamic range in that opening section, down to 0.01% impurity detection using laser-induced fluorescence, and industry-proven RSD is less than 3%, right? Again, those are part of the intercompany collaboration studies that were done. On the right, you see all of the validated application kits. So CE-SDS for either molecular weight or IgG purity, the CEIF, and the upcoming fast glycan labeling analysis kit. And obviously, there's a number of instrument features that make it robust and reproducible: liquid capillary cooling, sample temperature control, the pre-assembled capillaries, capillary cartridges that we talked about, and those biopharma intercompany collaborations that really prove that this thing actually works as advertised. And then modular detection, you can choose between UV, PDA, and LIF detection. So again, this system is really what enables us to make those changes and optimizations to run your assays faster and continue to get quantitative data. Now, I just want to take one quick second and mention that if you are at all interested in mass spec detection, we do have a system for that as well. It's called the CSI 8000 Plus. It performs all of these applications, so it can basically do everything the P800 Plus can do with LAF, UV-Vis, or PDA detection, including all of the fast applications that we talked about today, but it's been developed specifically to utilize MS detection. So if you have a need in the future or right now for MS detection coming from your CE, the CSI is a good choice there. Wrapping up on my last slide just with thoughts about the 3 main applications that we talked about. So we started with this idea of fast but quantitative analyses using CE. We went over fast glycan labeling. It can be up to 5x faster than standard HILIC methods with sample prep in under 60 minutes and separation and identification at the same time automatically in the software. Fast CE-SDS method, 2x faster than the standard method, again, separations in 15 minutes. You can run 2 times the amount of samples with our high throughput tray setup, and ease of use with our pre-assembled capillary cartridges. And then fast charge heterogeneity with CZE, 2x faster than CEIF. You get separations in about 10 minutes. Very easy sample prep. It's basically just a dilute and shoot with water. And simple method optimizations and flexibility if you have some particularly challenging molecules. So with that, I think we've got one final poll before we close and hand it over for questions. So the last poll is, are you interested in implementing any of these CE applications in your laboratory? yes within certain time frames, or no, not at this time. We'll give people just a couple of seconds to answer that one. All right, I think we can close up this poll, and we will move on to the Q&A with Dr. Mark Lees. I want to thank everybody for your attention today, and obviously if you have questions, we can answer them now. If we don't get to any of your questions, we will absolutely reach out to you offline and make sure that we answer them there. So thank you, everybody.
Thank you so much for that outstanding presentation. I just wanted to, before we get started with our question and answer session, to remind the audience of how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We're going to try to answer as many questions as we possibly can. I'd like to introduce Dr. Lees, who will be answering today's question.
Hello, can you hear me?
Lees? Very well. Thank you very much, Doctor.
Yeah, okay, great. I'm here, yep.
Perfect. All right, our first question is the data quality of the FAST CE-SDS option you mentioned similar to the regular kit method I now use?
Yeah, so the quality of the data that you get is going to be very similar to the and this is specifically, I think you mentioned for the FAST CE-SDS assay, correct? It's gonna be similar. It will be actually in about half of the time that you're able to get results if you're using the long side currently of the capillary. There may be a slight decrease in resolution between some of the very, very closely migrating peaks, but you should not see a significant decrease in quality of data. In terms of reproducibility, you shouldn't see a difference there at all.
Thank you. And for the high throughput tray setup and CESI analysis, do I need any different reagents that come currently with your kit?
For the high throughput tray setup, you can use the existing reagents that come with the kit.
Thank you. Who developed the CZE method for charge heterogeneity?
So the CZE method that was discussed in the presentation was actually an effort by a number of biotech companies. So initially, some of the work was performed by Genentech and published about 10 years ago, and then refined over the last 5 years, beginning with work done by Pfizer and then by a larger group of biopharma researchers. And ultimately, the technique that was described in this discussion was the outcome of that work as you can actually read about some of the details on that work in the publication that Matt referred to in, I believe it was one of his first 5 slides or so, where he actually discussed these intercompany collaborations. So that work is detailed in that particular publication.
Thank you. And our next question is, what kind of capillaries are used for N-Glycan separation? Are they standard bare fused silica or coated?
Right. So in the original assay that we currently have on the market, and this is the oligosaccharide labeling and analysis kit, we actually recommend using the N-CHO capillary, which is essentially a PVA-coated capillary, which really helps in minimizing electroosmotic flow and interaction of any analyte with the wall of the capillary itself. The way we have developed the FAST glycan assay is is to essentially allow, using the optimization of the chemistry, to essentially allow you to use bare fused silica capillaries. And that's why we're also including the pre-assembled cartridge, which is essentially a factory-built, pre-built cartridge with a capillary already inserted, cut, and windowed. And that capillary is a bare fused silica capillary. So we've optimized that assay using a bare fused silica version of the capillary.
Our next question, with the fast CE-SDS method, does it reduce the condition time of the capillary?
The conditioning time of the capillary is not changing, and so you're essentially you're still required to do the occasional conditioning in the method. The thing that changes there really is the separation time, because you're going from right to left in the capillary as opposed to left to right, which is the longer path of separation.
Thank you. And our next question is, I have an antibody that binds glycan, but I don't know which species it is. Is it possible to determine that?
Well, it depends. So I think the answer there is yes, it's possible to determine that. There's a couple of caveats, though, depending on. So one of the things I would probably need to follow up on is what sort of a glycan does your antibody bind? Is it an N-linked oligosaccharide? Is it some other kind of a glycan? Is it a monosaccharide, for example? So there may be some optimization necessary, or there may be other methods which are going to be better specifically for that species. The assay described here was specific for cleaving N-linked oligosaccharides from the peptide backbone of the antibody using the enzymatic digestion technique, and then labeling those sugars. So there may be some optimization that can be performed in this situation, but without more information, it'd be very difficult to make that to confirm that here in this setting.
Well, thank you very much for that. And I'll be wrapping up today's session here with some final comments. Matthew, do you have any final comments that you would like to add?
I would just like to thank everybody for their attention, and obviously if you have any questions on these applications, I know a number of you are doing CE-SDS or glycans today. If you have any questions on those applications or learning more about applications you're not currently doing, please reach out to us.
Thank you. And Dr. Lees, do you have any other final comments?
No, I think I'm okay. Hopefully the questions that were answered were insightful, and as Matt stated, we'd be happy to answer any other questions offline.
Well, thank you so very much to you, gentlemen. I'd also like to thank our sponsor, SCIEX, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through April of 2017. You will receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Until next time, goodbye.